Hello, my name is Nadav Ahituv, and I'm a professor at the Department of Bioengineering and Therapeutic Sciences, and also the director of the Institute for Human Genetics at UCSF. And we are really excited to be showing you what we're doing for the CRISPRa Research Foundation. And this is a project where we sort of aim to show proof of principle of a potential therapeutic approach for CRISPRa. And so what I thought I'd do today is just show you a few slides on what we're doing and then hand it over to Deanne, who's a graduate student in my lab who is working on the project to provide you more information. So with that, I'll share my screen and start this PowerPoint that hopefully you could see. And so our lab a few years ago decided to target for therapy diseases that are called haploinsufficient. Haplo is half and insufficient is just half is not enough. And so what does that mean? So we usually have two copies of a chromosome, one from our mom and one from our dad, and that provides us normal gene levels and everything's fine. Every so often, we could have a deletion in one copy of a gene, for example, here, and this gene is not functional, and that leads to 50% of the protein being made. For some genes, this is actually fine, but there are many genes where this could lead to disease. And when we look, for example, in the literature, there's over 660 genes that when you have half, it's sort of like running on empty, and you have human disease. And the disease could be quite devastating, including severe epilepsy, cancer, polycystic kidney disease, and many others. So a few years ago, and you've probably heard, CRISPR came out, and what CRISPR does is it can go and cut the DNA and make changes to it. What we've done here is actually use a different form of CRISPR where the scissors are actually mutated. So the scissors don't work, but you can use CRISPR as a delivery truck to bring things to a specific location in the genome, in the DNA. And what you could do with that delivery truck is connect things to it. For example, what's called a transcriptional activator, something that will make a certain gene, for example, make more RNA and more protein, and that's been called CRISPR-A for CRISPR activation. So going back to this example of the therapeutic approach, where we have one gene that is still fine, it's just giving us 50%, what we thought we could do with CRISPR-A a few years ago is target that existing copy and force it to squeeze out more RNA and more protein, and that could rescue these diseases that are called haploinsufficient. And it beautifully worked in a paper that we published about 4 years ago in Science. You can see here that we managed to rescue, this is just one example of two of the top obesity genes in humans, where you can see this is one copy, this mouse has one copy of this gene on the left, and you could see is extremely obese, while the mouse here in the middle also has one copy of the gene, but because we upregulate that existing normal copy, we get mice that look exactly like the ones on the right that are normal and are not obese. And so through that work, we showed that this could be a potential therapy for these haploinsufficient diseases. So how does that basically connect to CRISPRa? So going into CRISPRa, as you all know, there are two chromosome 5 copies, and one of them in the patients, as in the individuals, has this region deleted. And that region can have anywhere between 73 to 98 genes. And so this is basically exactly the same approach where we have one copy that is fine and one copy that is missing. However, here what we need to do is upregulate many genes and not just one. And so for our project, what we are doing is starting with one gene called CTNND2, which is thought to be one of the major genes leading to a neurological phenotype. And what we're doing for this gene is developing exactly what I showed you before for the obesity, trying to make an activator that will activate CTNND2 and make more of it, so like it would be that you have two copies, and that hopefully can help here. If that works, then we hope to develop this even further and try to activate not just that CTNND2, but also additional genes from the 73 to 98, and focusing on the genes that are most important in terms of the phenotype for Cri-du-Chat. And with that, I'm going to pass it on to Deanne, a postgraduate student in my lab, who will continue. Thank you.
Hi, my name is Diane, and I'm a graduate student at the University of California, San Francisco, in the lab of Dr. Nadav Ahituv. I have the pleasure today to expand on Dr. Ahituv's presentation. I will be sharing more information on the CTNND2 gene and experiments we're conducting in the lab. I would like to begin by thanking the families and advocates of the Cri-du-Chat Research Foundation, who without their support, trust, and contribution, the work I will be presenting today would not be possible. Second, the Cri-du-Chat Research Foundation for their funding and support. And finally, Dr. Nadav Ahituv, whose scientific leadership made this work possible. As you know, Cri-du-Chat syndrome, or 5p- syndrome, results from a deletion of the petite arm of human chromosome 5. As Dr. Nadav Ahituv mentioned, there are many genes in this region, and one of those genes is known as CTNND2, the gene towards which we have chosen to direct our CRISPRa therapy towards to. You might be asking, why of all those genes are we going after CTNND2 first? Well, there are three main reasons why we selected CTNND2 as our first target. First, as you might remember for Dr. Nadav Ahituv's presentation, this gene is haploinsufficient, meaning when one copy of the gene is not enough. In this case, when CTNND2 is one of those copies if deleted, there's not enough of that gene that is producing protein, and therefore the gene level is like running on empty. So you don't have enough protein to do the function, the normal function of this gene. Number two is the clinical relevance for Cri-du-Chat syndrome or 5p- syndrome. We want to go after a gene that actually has a phenotype that is important for families and patients with Cri-du-Chat to treat. And in the case of CTNND2, previous research has proven that CTNND2 is in part responsible for a lot of the neuronal phenotypes we see in Cri-du-Chat syndrome. Third is previously known gene function. Thanks to many scientists before us, we know a little bit more about what CTNND2 is actually doing. So CTNND2 is actually almost exclusively expressed in the brain, and therefore it's mainly only present in the brain. You might not know this, but neurons are the cells that make up their brain, and I think they're fascinating. Why? Because they have a really interesting structure. These neurons must have an area of the body that contains the nucleus, and in the nucleus we find the DNA. But neurons must make really long connections in the brain, so they have this really long projections, and they use this end part of the cell to make connections with one another. So now that you know a little bit about the neuron morphology, let's talk about where we find CTNND2. CTNND2 is found in two main places in neurons, and that is in these extremities that cells use to make connections with one another, as well as in the nucleus, where the DNA is found. So CTNND2 in those these extremities, it acts like a glue. It's found between two cells. If we zoom from here, it acts like a glue in these extremities, and it's important in a process known as cell adhesions, where cells make connections with one another to transfer information, nutrients, and many other things. In this part, CTNND2 is important for one, cell adhesion, and also a process where cells migrate, where they're able to connect signals as well. The second function of CTNND2 is actually in the nucleus, where it binds to specific genes and actually activates that gene expression. This is known, all this information I've shared here is known previously from studies in mouse models, as well as studies in cells. Now that we know a little bit about CTNND2, what exactly are we going to be doing in the lab? Thanks to the CRISPRa Research Foundation, we obtained patient cells. A technique that I've learned previously is that we can use is where we add to this patient cells certain factors that actually makes them be neurons on a dish. What we can do is with this patient cells, is add our CRISPR-A therapy that has been developed in the Ahituv Lab previously. But something that cells are very good at is not letting other external factors in. So one challenge that we face is how do we get this therapy into the cells? We use AAV or adeno-associated viruses to actually act as a delivery mechanism. So if you think of SDCas9 as a delivery truck that takes our therapy to the gene of interest, AAV is like Amazon. But in the time of COVID especially, virus may seem like a scary thing. So why exactly are we using AAV as a delivery system? Well, just as a reminder, AAV stands for adeno-associated viruses. And in fact, they're the leading platform for gene delivery treatment for many diseases. Here, I have outlined a timeline which I adapted from a review that was published in 2019 in Nature Reviews. And what we see here is selected milestones in the adeno-associated virus gene therapy development. What we see is that we had the discovery of AAV in 1965. Why is this important? It's because we discovered AAV in the 1960s, which has allowed many years for the efficacy and safety to be established by other researchers. The AAV vectors to treat a human patient, the first human patient that was treated was in 1995 for the treatment of cystic fibrosis. In 2008, the efficacy of AAV was further tested in a congenital eye disease. And finally, in 2012, two other key dates that I want to emphasize here is the approval by the European Medicines Agency, as well as the approval of the Food and Drug Administration safety in 2012 and 2017. These are key dates because it was the first gene therapies to be approved by safety agencies in Europe and the US. What I really trying to convince you here is that AAV is a safe and efficient way to deliver many gene therapies that we're taking as an advantage to develop the first therapy for Ahituv syndrome. Thank you so much for the time, and I hope I've shared a little bit more information on CTNND2 and AAV.